Well, good evening, and welcome to Lightning Talks. I'm Jim Wharton. My pronouns are he and him. I'm the Vice President of Conservation Engagement and Learning here at the aquarium. Now, we're coming to you live from the Seattle Aquarium on the shores of Puget Sound in the contemporary and traditional territories of the Coast Salish people. And normally, it's just like me and Becky and Megan and Michaela huddled around our computers, broadcasting out to all of you. But tonight, we have this amazing live studio audience. So we're so thrilled to have their energy. We're really happy to have you all joining us as well. And we have a really special program tonight. And actually, tonight's a really cool kind of special night because tonight is the 20th edition of Lightning Talks. I can't, but, and I look so young, right? We've had in 20 editions 141 speakers who have delivered exactly 705 minutes of content. And it's possible we went over here and there, so maybe it's like 710 minutes, but 5 minutes apiece, quite a bit of content. Now, we've had speakers from all over the Puget Sound, all over the world, but can anybody hazard a guess as to which institution has been represented most often in Lightning Talks? I heard it. University of Washington has had 44 speakers in our 20 Lightning Talk events, including 2 speakers tonight, Jan and Eileen from UW. We've had many great partners, NOAA, of course, the Seattle Aquarium, and many others that have come together to share the science of the ocean, the conservation priorities of the ocean. It's been really cool. In fact, we've had incredible variety of topics and presentations. We've had a lightning draw. We've had a lightning dance. We had climate haikus. We've had talks on the blob, on orca poop, on mud minnows, on narwhals, on Fukushima, and even like sushi parasites. So it's been quite a journey. And fun fact, I was not the original Lightning Talks moderator, but I was our very first Lightning Talk speaker. So it's been quite a while, and it's been quite an assortment of topics. But can you guess? Actually, let's take a second. I want you to turn to the person next to you, see if you can guess the animal or group of animals that's been most represented during Lightning Talks presentations. Take a second. And if you're online, drop those ideas into the chat. We'll see if you can figure it out. All right, well, if you guess sharks, then you were right. So we've had 16 different presentations on sharks, followed by otters and whales, and a whole bunch of other things. And for those of you who know me, I mean, maybe it's not such a surprise that sharks came out on top, but I would just like to take credit for only 16 presentations out of 141. That's what you call restraint. Now, what we're going to do now, though, is we don't really have time for showboating megafauna tonight, because tonight, we're talking about snails. Now, this is my kind of audience. Now, snails was an audience recommendation. So if you all have ideas for what kinds of topics and themes you'd like to see, please do share those with us. Snails is directly from you all. We'd love to hear what else you have in mind. And of course, as usual, we have 5 great speakers to tell you more. So if you've never been to Lightning Talks before, here's what you have in store. You've got 5 speakers with 5 minutes each to share one big idea and not 1 second more. Now, they'll get warnings at 1 minute. They'll get another warning at 30 seconds. They'll get a final warning at time. And if they go over, they'll get this. It's real. Now, it's if they get it, it's no big deal. It's kind of fun, but at the same time, it's definitely something that I've heard there's minor anxiety about. But so we will not be stopping after each question, or excuse me, after each speaker for questions. We'd like to keep the whole thing moving, but we are going to do a Q&A conversation at the end. So if you have questions throughout the night online, please do put those into the chat. We'll be collecting those throughout the evening, and we'll bring those back to our Q&A conversation. For the folks in the room, they're going to be writing their questions down on notepads, or be raising their hands and asking questions live. So we'll work both of those into our evening tonight. Now, if you like programs like Lightning Talks, please do consider a donation to the Seattle Aquarium. When you donate to the Seattle Aquarium, you're joining us in our mission to inspire conservation of our marine environment. But that's enough from me. It is time to get our show started. And it's really our pleasure, and there's no better way to start the show than with the Seattle Aquarium's own Dr. Erin Meyer. Now, Erin is our VP of Conservation Programs and Partnerships, a mycologist by training, and the exact right way to start the show. So, Erin, are you ready to go?
As ready as I'm gonna be.
The floor is yours.
Fantastic. Hi, everybody. Thank you for being with us here in person and online. I'm Dr. Erin Meyer, and I'm here to kick you off with the wild world of snails that you never knew existed. So snails are everywhere. Then they have been radiating around the world for over 500 million years. There are over 40,000 known species spread all throughout the habitats and the oceans, on land, and in the deepest parts of the ocean. 40,000, that's 8 times the species of mammals on this planet. And snails come in many, many shapes and sizes. The smallest one is the size of a tip of a ballpoint pen, just recently discovered at 0.03 inches. The largest is over 3 feet across and comes in at 40 pounds, so it's the size of my 5-year-old son. But snails, while they've been around for a long time, some of them are going extinct. And actually about 30% of the gastropods, so slugs and snails that have been assessed, are listed as threatened, so critically endangered, endangered, or vulnerable, and counting. But we also are connected to snails in many, many ways. We eat them. They're really good with lots of garlic and fried and even raw. Maybe too good because they get overexploited. We also trade with them. And what you see pictured here, this is an indigenous tribe from Papua New Guinea. Those rings you see in the back, those are rings made of Nassarius snail shells. So that's a symbol of wealth. And that young woman you see on the right, we also wear snails. So that is a young woman from the Taloa Dani Nation, and you see she's wearing a necklace made of snails as well. And indigenous tribes have been making regalia out of snails for a really long time. And you can also get them yourself at the stores, you can see with my earrings, maybe. Now, what do snails eat? We know we eat them, but what do they eat? Well, snails have this modified structure in their mouth, which is a toothed tongue. You can see it there in the center. And in this particular snail, they use it to scrape. They can scrape on leaves. They can scrape algae off of rocks and rivers and in lakes and in the ocean. They can also drill holes, so that radula can be modified to look like a drill bit and drill holes into shells like oysters. You're going to hear more about a species that does this later. They also stab. So cone snails have a modified radula, and they stab things like fish. They also snack while surfing. So this is an olivid snail. Using these with permission from, you know the guy who pulled a straw out of a sea turtle's nose? Well, he also really likes to take videos of snails, apparently. And that picture you saw on the left was snails surfing around, an olivid snail that eats other olivid snails that also like surfing. And I mean, who doesn't like to snack while surfing? They also use fishing nets. In this case, the fishing net is made of mucus. So they use these, let's just say, sticky booger nets to catch their prey at night. So that's fun. They use tools. Okay, now I'm bending the rules a little bit, but snails, let's just say gastropods have lost their shells many times, so I'm sneaking this one in. These are sacoglossans. They are solar-powered sea slugs. They steal chloroplasts from algae, and they're really awesome, and that amazing leaf sheep you see in the middle is probably going to be the cutest animal you're going to see all night. Okay, so how do snails move? They move in a lot of ways. They crawl and climb, probably what we most often think of them doing, but they also go rafting. So they make rafts out of bubbles and float upside down their entire lives. They also swim. These are pteropods. You're going to hear more about these from Jan in the next talk. So it's a snail's world. They live everywhere, and there are loads of them. And, you know, we're connected to snails in so many different ways, for culture, for medicine, for beauty, for food. You know, they eat everything with all sorts of different varieties of radula that drill and poke and scrape and all sorts of things. And, they just kind of do all the things. And I didn't talk about this, but they can also hibernate and estivate so they can sleep through hot weather and cold weather. So, again, in conclusion, snails are just awesome. And so just kicking us off with that, thank you for everyone for being here, for listening, and, because snails are important for sharks too. So thank you.
Thank you, Erin. I'm not mad that you snuck a shark into the end there.
I sure did.
So, you've got 40,000 species of snails, and I'm going to make you do it. What's your favorite snail?
Okay, so I'd have to show my first slide. It's Satyrium pica, which is the West Indian top snail, and actually has 40 different common names. I did my dissertation on that species, and it's awesome.
Well, that's amazing. Thank you so much, Erin. Let's give her a round of applause. Now, if you have questions for Erin, please make sure that you're putting those into the chat, and folks writing those down on your pads or noting them on your phone or however you want to do it. Erin will be back later during our Q&A. Now, our next speaker is Dr. Jan Newton. Jan is a senior principal oceanographer at UW's Applied Physics Laboratory and the co-director of the Washington Ocean Acidification Center, among a huge list of other titles, as long as my arm. But she is also a Seattle Aquarium Research Award winner. And tonight she's going to tell us a little bit about those delicate swimming snails that are almost quite literally canaries in the coal mine for the impact of climate change and oceanification on our world ocean. Jan, are you ready to tell us more?
I was born ready, Jim.
All right, take it away.
Thank you so much, and it's great to be talking to you, but I have to warn you, this is a bit of a sobering reality check, because this story is not just a super happy one. Pelagic pteropods means that they're in the open sea, and I'm going to tell you about how they're giving us some information about ocean acidification. All right, but first let's just dwell on the beauty of these pteropods. Sea butterflies, sea angels, some have shells, some don't, but I think we can all agree, very beautiful. Now, here's a couple of textbook examples of marine food webs from the pelagic zone, and look at that, pteropods in both of them. So, what do we take from that? Very important. All right, this is a cool one that I didn't actually know before I put this talk together. This shows where pteropods started to inhabit the earth. We are a freaking newcomer to this. We're off the scale there. Pteropods are very ancient. Okay, so now let's look at this. There's two pteropods on there, and they look different. The one on the left looks pretty normal, and the one on the right has those stripes. And what that is their shell starting to dissolve, the dissolution, to corrode. And so what we know from this research is that they are very susceptible. Okay, what is causing this? Ocean acidification. What does ocean acidification mean, or OA as we call it? Carbon dioxide from the air is added to seawater, and that changes the seawater chemistry. It changes the chemistry of the seawater. Reduces the pH, that's why we call it acidification, and it also locks up the availability of calcium carbonate, or what the pteropod use for their shells is aragonite, all right? So there's other processes that affect carbon dioxide in the ocean, and here's just a beautiful schematic that shows that photosynthesis takes up CO2, but then respiration releases it, just like you all are doing now. A marine food web is doing that. So when we go out and look at seawater, we see both a signal from natural processes and from ocean acidification, and that is exactly what we do with the Washington OA Center. We go out seasonally on the Rachel Carson, maybe you've seen her out there, and collecting water chemistry samples, plankton samples, and then trying to understand what the Salish Sea is telling us. All right, so here's that pteropod, very delicate shell, and its ability to maintain that shell is due to the amount of aragonite in the seawater. So these are data. I know I'm showing you graphs and I'm not supposed to do that, but what I want to tell you is that on the bottom part it says if you go in that direction, that's bad. And so the stations and the data are color-coded, and you can see there's 3 categories: one from the Strait, one from the main basin in South Sound, and one from Hood Canal and Whidbey Basin. And it's that latter, purple category, that's the most severe, most corrosive. And that one is a kind of a benchmark for where the shells will start to dissolve. All right, so Nina Bednarsik, Dr. Nina Bednarsik, who's now at OSU, has done a lot of research, and she was able to take the shells and figure out type 1, type 2, type 3 dissolution, type 3 being the worst. And you can see even one of them has a hole in it. All right, that's not good. All right, so here's another graph, but it's an easy message. The bigger the dot, the more dissolution in these samples. These are from 2014 to 2016. We'll have more data coming soon. The bigger the dots are in the purple orb, that is where Hood Canal and Whidbey Basin are. Okay, here's another data graphic, and it's showing you some of the data. And what I want to show you is that if you take the middle part of those, it's around 20% dissolved, but that middle graphic was 2015. That was our blob year, and it goes from 20% to 30%. That's not good. So temperature is another stressor. And lastly, what I want to show you is that pteropods are also actually revealing something about their life cycle, and this surprised us that the ones that were the most severely dissolved were in April, in spring, and that's because they actually overwinter and then reproduce in the spring. So what we really want to do is be listening to these pteropods more. They've got a lot to tell us, and it's really important. So thank you.
Thank you, Jan. So one of the things I'm wondering is that those pteropods, they are so delicate. Even the healthy ones seem almost translucent or transparent. How much acidification would we need to see before it starts really damaging some of the hardier snails that we would think of normally?
Hello, there we go. We are seeing that damage, and so we're seeing it in the pteropods, but those are the sensitive ones. But we are also seeing this kind of effects on things like oyster shells, and it's not the hardy oyster shells, it's the larval, and they deposit the aragonite into this little thing. It looks like a D. They call it the D-hinge shell, and those can be deformed by current day conditions. And the conditions, I hope you got that from the data, they vary drastically with location, with depth, with season. And it's really understanding where are most vulnerable populations and where are they resilient to help understand how we can best protect, and we gotta curb those carbon emissions, just bottom line, period.
Well, thanks again, Jan. Jan will be back for our Q&A session, so don't forget to put those questions into the chat, and I hope folks are writing down a few questions for Jan as we speak. Now, our next speaker is Julia Sigwart. She is the head of malacology at the Senckenberg Research Institute in Frankfurt, Germany. Julia is about to tell you about a snail, the likes of which you have never heard of. That is a full-on guarantee. Now, Julia is very sensibly sleeping right now in Frankfurt, Germany. It's about 3 a.m., but she is beaming her talk from her subconscious to our Lightning Talk servers just so she could be here tonight. And so, Julia, whenever you are ready, the floor is yours.
Hello to everyone in Seattle. It's my absolute pleasure to be able to talk to you this evening about one of the most extraordinary snails in the world. This is the scalyfoot snail, Chrysomelon squamiferum, and I'm Dr. Julia Sigwart. I'm a professor and curator at the Senckenberg Museum in Frankfurt, Germany. This snail, you might have noticed, not the shrimp, the snail, This is very different to any other snails on land or in sea. You can see that instead of having a soft, squishy foot, its foot is actually completely covered with hard scales that stick out like a porcupine in all different directions. But what's also extraordinary about this snail is that it was very recently, in 2019, the first ever animal that was declared to be globally endangered by the IUCN Red List because of the threat of deep sea mining. So just to orient you to where this amazing snail lives, it lives in the deep sea hydrothermal vents, which are geothermal anomalies that occur at the seafloor all over the world, usually at depths of about 3 kilometers or about 10,000 feet below sea level. You're probably at about sea level. So how far is 10,000 feet? Well, there's a couple of other things here to orient you. So Mount Everest, famously the tallest mountain on Earth, is about 5.5 miles high. Yosemite is about 4,000 feet high. The Grand Canyon goes down a little bit more than 5,000 feet below the canyon rim. But we're talking about depths that are much, much deeper than that, double that depth, but not nearly as deep as the ocean can go. The other thing that's on this chart, just to give you a little bit of orientation, is the depths of some famous and reliable research submersibles. So one is the American research submersible Alvin. These are human-occupied vehicles that are used for deep sea exploration. And the other one is the Shinkai, which is the Japanese research submersible. And that was one that I was very privileged to be able to dive in and study the scalyfoot snails in their natural habitat. So the habitat of the scalyfoot snails is underwater volcanoes, deep sea hydrothermal vents that look like this. There are areas of the seafloor where we have mineral precipitates that come out super hot and super fast, and all of these black smoke is actually minerals that are making contact with the surrounding cold seawater in the deep sea and then precipitating out. The scalyfoot snail is known from 3 main locations in the Indian Ocean. So this is the coast of Africa and Madagascar, and you see the 3 locations around Mauritius, the island of Mauritius, and then 2 others, Kai Rayfield, which is currently under a mining exploration license granted to Germany, and the Longqi field, which is currently under a mining exploration license granted to China. There are no conservation areas for the scalyfoot snail or for any hydrothermal vents in the international waters. So as I said, we were able to take the Japanese research ship Yokosuka and its amazing deep sea underwater research vehicle, the Shinkai 6500, in 2016, and dive to find the scalyfoot snails and do a lot of very important work and experiments on these amazing animals and observe them alive. And through part of this work, we were able to really get to understand the anatomy of the scalyfoot snail. What's most extraordinary about it, you can see a little bit of this here in this somewhat cryptic 3D model. So this is a reconstruction of all of the different organ systems inside the snail. We don't want to just think about the scales, but also all of the other adaptations that shape how this animal interacts with its environment. And what's most amazing about this snail is that all of these adaptations, including the scales, are actually driven by its relationship with bacteria. This snail doesn't eat. It has a huge organ on the inside, which is the kind of browny cloud in the middle of this model, which is where it houses endosymbiotic bacteria. And the snail carries around all of these bacteria in its big inside pouch, and the bacteria actually feed on the chemical energy of these geothermal environments, and that feeds the snail. And what we were able to show is that all kinds of other adaptations around the anatomy and physiology of this snail is driven mainly to make the bacteria the best possible home. But one amazing thing about the snail scales is that they're actually magnetic. So you saw these pictures before. The scalyfoot actually comes in different colors, and this is because of the different mineral composition of these geothermal environments. So when the vents have very, very high levels of iron, that iron actually gets incorporated directly into the skeleton of the snail. In this case, its shell and its scales. So here we can see that the scales have so much iron content, you can actually pick them up with a magnet. But this isn't really coming from the animal. This is mediated by the bacteria and then incorporated secondarily into the shell. So the white one lives in areas where there's much less iron that's available through the hydrothermal vents. But again, the saddest thing, or the most important thing to know about this animal, is that it's threatened with extinction. It's declared globally extinct because of the threat of deep sea mining. This is an issue that is very real, that is more and more present, I think, and people are more aware of it, and something that just adds to the list of many reasons that we have to fear how humans are impacting our environment and driving species to extinction that we share this earth with. My group in Senckenberg here in Germany is working to try to save as many of these species as possible, first by naming species that are yet undiscovered by science, and secondly by working with the IUCN Red List to make sure that we have as much information as possible about the threats to these extraordinary and vulnerable deep sea species. So we would be delighted if you want to follow us on social media and know how you can get involved and join our efforts and our alliance. Thank you so much for listening, and I hope that you enjoy this and all of the other wonderful creatures that you get to hear about tonight.
Thank you very much, Julia. Did that seem like more than 5 minutes? Yeah, I think she's on the metric system or something. So if you have questions for Julia, please do put those into the chat. We're going to add a couple of links into the chat as well to give you a chance to go learn even more about the scaly-footed, big-hearted iron snails. And I wish Julia could be with us tonight for Q&A, but if you have questions, let us know and we'll try to get some answers for you. Now, our next speaker is Eileen Bates. Eileen is a PhD candidate at the University of Washington, and she works with the Puget Sound Restoration Fund on a project to help restore the populations of pinto abalone to Puget Sound. And it's a project in which the Seattle Aquarium is very proud to play a key role. Eileen, are you ready to tell us more?
Sure am.
All right, take it away.
All right. Okay, so our next snail today is the pinto abalone. Some of you have probably heard the word abalone, maybe eaten an abalone, maybe have jewelry made of abalone shells. What you might not know is that we have a species of abalone right here in Washington, our Washington pintos. Unfortunately, because they're so tasty and because their shells are so beautiful, they've been pretty drastically over-harvested. So between 1992 and 2017, there was actually a 97% decrease in the number of pinto abalone in Washington. So my work with collaborators is trying to bring pintos back to Washington. So I'll talk to you about how we do that and how it's going. But first, setting the scene, pinto abalone live in kelp forests and rocky reef subtidal ecosystems in Washington. And they are vacuum cleaners of the seafloor, you could say. They roam around grazing on diatoms, other algae, and really clean up spaces for other species to move into those areas. And they're also an important prey species for mammals, fish, invertebrates, lots of things like to eat them. So they're really important for the natural environment. They're also really important to people. They're culturally and economically important for tribes in the region. And then they also supported a recreational fishery before it was eventually shut. So really important to people and the environment in general. But unfortunately, there are now so few adult pinto abalone, like the one pictured here, that they physically can't find each other to mate and rebuild their population, even though fishing pressures have all been removed. So this is where human intervention comes in. What we're doing is basically trying to bump up the number of abalone in the wild so it's back to a level where they can be self-sustaining. And how are we doing that? I'll get into the nitty-gritty of it, but I'll also tell you a little about the pinto abalone life cycle while we're at it. So we'll start with the adults. We go out into the field, so we dive out in Puget Sound, and we find these abalone all by themselves. We try to find them from a wide range of places, so we get a lot of genetic diversity, and we bring them back to our hatchery. And this is the hatchery that Puget Sound Restoration Fund runs over in Manchester, Washington. And this is where all of our animal husbandry happens for abalone. So Once we bring those adults back, the next goal is to spawn them, which can be quite a challenge. When we are successful, it's a pretty hectic day because we're trying to make as many crosses of different sperm and egg combinations as possible, so we maintain as much genetic diversity as we can. So after we spawn them, they move into these little hatching tanks, and at this point, they look like the guy in the lower left corner there, so not very snail-like. They're actually free swimming for the first week of their lives. And you can see this one is just starting to build its shell and is pretty vulnerable to ocean acidification, which we heard about from Jan. But after about a week in that phase, it goes through settlement, is what we call it. So it basically drops out of the water, metamorphoses into the adult snail that we recognize and love. And once that happens, we move them to these bigger tanks, these nursery tanks. We have about 98 of these over in Manchester, Washington, run by PSRF. And then we have 32 of them, actually, in the aquarium's new animal care facility, which is very exciting. And what's also exciting is we're in the middle of abalone spawning season right now, and 10 of these tanks now have new abalone in them. Some of them just arrived earlier today. Yeah. So it's a thrilling time. And while they're in those tanks, they're growing. They're very slow growers. They're trying their best. They're grazing on diatoms and other algae. And eventually, we will bring them back out to those rocky reef ecosystems. So what this looks like, we transfer them in PVC tubes, so we can fit a lot of them in a really small space and minimize our chances of crushing them accidentally. Let's go back. We dive out with them in mesh bags in those tubes into the ocean, and we nestle those tubes into their rocky reef ecosystems. From there, they can crawl out, keep eating, keep growing, and eventually start reproducing and bringing their population back. So, over the last decade or a little more, the collaborative people working on this project have put out almost 50,000 abalone, which is great. And they have put them at 29 different restoration sites. So what that means is they're all pretty concentrated in small spaces, and that's kind of the goal, because then we'll have a lot of abalone in a little space, and they can rebuild from there. And thank you.
Thank you so much, Eileen. I gave you an extra few seconds for all of the applause. I thought that was fair.
I needed it, yeah.
So, from my understanding, these outplanting sites are super secret, right?
Yes.
Like, they're not public knowledge. Why do they need to be secret?
Because the abalone are so tasty. So we don't want people to get any ideas. Don't look at any of the pictures too closely, not that you recognize anything. What's fun is that all the outplant sites have nicknames, and they're all named after amphibious Allied assaults from World War II.
Well, of course they are.
If you look up, the list, it's a fun list of names.
All right. Well, yeah, so please don't memorize any landmarks, and thanks, Eileen, again. So if you have questions for Eileen, you know the drill. Put them in the chat. Write them on your pads. She'll be back at the end with the rest. And it is time for our final speaker of the evening. Jen Strongin is a field outreach specialist for the Seattle Aquarium's Beach Naturalist program. And Jen is also an excellent and professional photographer, and I'm really looking forward to seeing some of her work in her presentation this evening. Jen is going to be telling us a little bit about one of the most charismatic members of our Puget Sound coastal community, and one that you can visit all summer long during low tide. So, Jen, are you ready to tell us more?
I am so ready.
All right, bring us home.
All right. Well, to end our evening, we are going to be talking about the mighty moon snail. Here in the Pacific Northwest, we actually have more than one species of moon snail. We have several. We have the Arctic moon snail, the pale moon snail. We have the Aleutian moon snail that you can see in this video cruising along the tide pool with its speckled foot. And that speckled foot is a really great way to differentiate this species from the one that I am going to highlight tonight, which I think is the mightiest of all of our local moon snails. This is the Lewis's moon snail. Maybe not as mighty as the large snail that Erin mentioned, that was the size of her 5-year-old, but these Lewis's moon snails are quite large. Their shell can be 5.5 to 6 inches in diameter, so imagine a large grapefruit, and they can extend their foot 12 to 15 inches beyond that shell. And that large foot contains some hollow passages that are called sinuses, and the moon snail can fill those up with seawater, and it does that to inflate that foot so it can use it for moving around, capturing prey, making eggs, and also burrowing into the sand like a plow. So here's a moon snail in West Seattle. This is the Lewis's moon snail, and you can see it's using that amazing foot to dig itself into the sand. You're going to find these animals in sandy areas. You're not going to find them in super rocky beaches. They need sand. That's where they find what they like to eat, and it's where they can burrow in and protect themselves. And you can see there's some nice mucus trail coming off the back of that. Mucus is so important to so many animals in the intertidal, including these snails, not only for getting around, but it's a key ingredient for making their eggs. At the bottom of that foot, there's a hard part called an operculum, and it's attached to the foot. It's kind of like their front door, so they can expel all that water that they have used to inflate their foot, and they can tuck that entire massive foot and mantle back into their shell and shut the front door. So that protects them from animals that might think they're delicious, like octopuses or crabs, or maybe birds if they're out and exposed at the low tide. So if you're out on our local beaches and you're looking for these snails, finding them isn't always easy. They're not usually above the surface. They like to be under the sand. But there are some clues you can look for, including shells that look like this. You saw some of these earlier in Erin's slide. So the Lewis's moon snail has that amazing organ, the radula, which is like a tongue with hundreds of tiny sharp teeth that they use like a drill to drill into these clams, and then they're going to excrete a digestive enzyme and slurp it all up. And so you can find these shells on the beach. That's a good clue to look for. And this is another clue. Some of you may have seen these on our local beaches. They're often mistaken for garbage. I've come across many a well-meaning beachgoer who is picking up beach trash and has a bag full of these. But these are the egg collars that these amazing animals make. And the female will gather sand on her big foot, and then she extrudes this jelly mass of eggs on top and adds some more sand. And then here's where that wonderful mucus comes in. She's going to seal it all up with mucus and then create these amazing structures. And each one can have about 500,000 eggs inside, which I think is so crazy. People always get excited about that on the beach. Those eggs are going to hatch out in a matter of weeks, and then these egg collars will disintegrate. So this is a moon snail I saw just this week. This was over in West Seattle. It had its foot out. It was cruising along. You can see that it's got some things that look like antennae. Those are their sensory tentacles that they use for smelling their way around and finding a mate. And there's also a siphon that you can see sort of underneath those on the bottom, and that's what they use for taking in seawater to get oxygen, or to get water to go across their gills and keep them oxygenated. They live quite a long while. They live about 14 years, and you can see this one maybe have been around for a while. It's got lots of barnacles attached to it. And then when the animal is no longer in their shell, when they've passed away or been eaten, these shells make a great home, not only for barnacles, but also for hermit crabs and for sometimes octopuses. Someone found an octopus in a Lewis's moon snail shell one time at the beach. So you never know. It's why we love to leave these shells at the beach, because they make homes for lots of other animals. And that's it. Thanks, everyone. Thanks for letting me talk about them.
Thanks, Jen. So if somebody wanted to come see a moon snail on one of our beaches, what are some of the good beaches where the beach naturalists are at during low tide where they might see a moon snail?
Oh, some great beaches to see moon snails in Seattle are Golden Gardens, Charles Ritchie Viewpoint in West Seattle, Seahurst Beach a little bit south of us. Dash Point's another great one that has lots of sandy substrate. So those are some great beaches to go to if you want to find some moon snails. We've saw lots and lots of moon snails this summer.
Well, thank you so much. If folks would like to know more about the Seattle Aquarium's Beach Naturalist program, please do come to our website. You can see the list of low tide days where you can find the naturalists out on the beach. Thanks again, Jen. Jen is going to be coming over to the stage now, and we're going to transition to our Q&A conversation. So if you haven't had a chance to yet, please do. You still have plenty of time to put questions into the chat. I've already got a long list to start from, and of course, in a few moments, we'll be asking for questions from our audience as well. But as we're organizing ourselves, I'm curious, do any of the speakers have questions for each other? I do. Yeah. Yeah.
No. Okay, here we go.
There we go.
Thank you. Jen, I've always been fascinated by moon snails. I grew up over, West Seattle, Lincoln Park, all of those places. But what fascinates me is how that outer part of that collar is so smooth. How do they get it so smooth?
It is very smooth, and when they extrude their eggs, it goes in between their shell and part of their body. So I think that is what causes it to be so smooth, and it's formed around that shell, which gives it its shape. And of course, that mucus that's sealing everything together makes it also very smooth, but it's being compressed as it's all being extruded. So, yeah.
How many weird conversations have you had on the beach about toilet plungers?
So many.
So I've got a couple of questions. People are very interested in the way snails work. So, Erin, like we see snails in the water, we see snails out of the water. Do snails belong in the water, or do they belong out of the water? And if so, can they breathe in the water, or do they have to come out of the water to breathe?
They belong everywhere. They've been here for over half a billion years. So they're everywhere. So when they're on land, oh no, there's exceptions to every rule. There are snails that have lungs. There are snails that have gills. There are snails with gills that kind of hang out in like the rocky intertidal. So if you've been down to the intertidal, you've probably seen snails kind of high up in the intertidal. Those ones still have gills. And they hold water inside of their shells that they can breathe some oxygen out of while they're hanging out outside of the water, or just kind of hold their breath for a while. So they kind of do it all.
So, Jan, we had a question about the map that you showed. And so there are some areas where you're seeing much more intense, the dissolution and other channels. So why is there so much variability in the CO2 levels in various spots in the Sound?
Yeah, that's a great question. Yes, why Woodby Island and why Hood Canal? So most people look out at Puget Sound and they see blue water, and that's about it. But an oceanographer looks out at it, and by taking some samples of the water and measurements all the way down from the top to the bottom, we see structure, because some water is what we call stratified. The warmer and fresher waters up at the top, and the colder and saltier waters down at the bottom. And that stratification can be both a good thing and kind of a bad thing. The good thing is that when phytoplankton start to bloom, they don't get washed out like they do in the main basin, which is really well mixed, okay? And the strait, also really well mixed. So it takes a lot of sunlight, a lot of nutrients to get those blooms happening. But in places like Hood Canal and Woodby Basin, the phytoplankton start blooming like crazy. Well, that's a good thing if you want to eat phytoplankton, but when that phytoplankton falls out, or the critters' fecal pellets who are feeding on the phytoplankton, all of that detritus sinks out, and it's in the deep waters, and then the bacteria break that down, and that's the respiration, releasing CO2, just like your compost pile happens. And so that's why you get more CO2 down at depth. So that's what makes those basins different.
So a lot of variability in environmental conditions throughout Puget Sound, because it's a very complex body of water.
It really is, and I use two descriptors of Puget Sound, diverse and dynamic. And so while Hood Canal is going to be typically much more stratified than the main basin, dynamic in the sense that other things can happen, storms and what have you, or high rainfall year, low rainfall year, and those things can vary quite widely. And so that's why long-term observations are really helpful, because they help us to see what patterns are ephemeral and what patterns are more variable, and just really helps us to see an environment for what it is, because most people don't think of water as an environment. You think of the rocky intertidal or the sandy beaches or the eelgrass beds or kelp beds, but water is habitat, and it has structure and it has all this variability. We just can't see it with our eyes.
So there's a question that's related to sort of conditions around abalone. So what are the conditions that are required for spawning? Like, what triggers it, and how did you all figure that out?
Oh, that's a good question. In the hatchery is different than in the wild. So in the hatchery, we're trying to get a bunch of them to spawn right at the same time during the day while we're there, and we can mix the eggs and sperm as we want. So we actually put the abalone in hydrogen peroxide baths to spawn. So they'll sit in there for a while, get a little uncomfy. We put them back in the water, and normally that's sort of enough of a uncomfortable shock that they will spawn. I say normally, but that's not actually true. It's very hard to get them to spawn. They're really resistant to doing it. So it's a challenge. But yeah, abalone hatcheries, so a lot of places in the world, abalone are not endangered, and they are farmed, much like we farm oysters here. So abalone, or using hydrogen peroxide to spawn, is pretty ubiquitous across all abalone species, and we're just doing it for conservation, where most groups are doing it for economics. Yeah.
So, Jen, someone was asking about the moon snails in your photography. Are moon snails a common feature of the things that you take pictures of? What is it about a moon snail that draws you to them?
Oh my gosh. Well, I just feel like they're like a lot of snails or invertebrates that have no face or head. They're just underappreciated. And to me, I just think they're so beautiful and fascinating. And I love the shape of their shell, and their foot has so much incredible texture, and just the fact that they're out there plowing through sand, they just fascinate me. So I really enjoy photographing them, and I don't get to see them very often either, because they're often under the sand. So I get very excited when I do get to see them, and I really enjoy taking pictures of them.
It's one of the things I like about programs like this, when we get to feature an animal that's not a shark, which are also cool, but people get to see new things about animals that they think maybe they knew some stuff about. Like I was super excited to put this program together, and the scaly-footed iron snails were brand new to me. That was I'm like, oh, wow, we need to talk about this. And it's the variability, like Erin, you said 40,000 species, and species are all unique, but there's such incredible variability. One of the things you didn't talk about is, are snails smart? Someone's asking, can you train a snail?
Yes.
And if you can, why would you want to?
So yes, you can, and people have. Why would you want to? I don't know. That's a great question. Well, I think the people that I know who have done it are usually little people, so children who are training them to go to a certain place to eat. I've seen videos of people who have trained snails to do a particular activity, like crawl up a certain thing, or they do jump, or to jump. They're not gonna, I don't know. They can't do the same thing that your dog can do, or that maybe even your cat can do, but they can do cool snaily things. But yeah, I've never personally trained a snail, but I know it's possible. So yeah, they're smart.
Yeah, life goals, right?
Life goals. I guess I'll do that this weekend.
Yeah, snails, so we love snails, but snails are not always the good guys, right? Are there not invasive snail problems? Does anybody know anything more about invasive snails that they can share?
Yes. Okay. So So there are definitely invasive snails all over the world. It's one of the reasons why, if you've ever looked, I guess back when they used to give you a paper form when you were coming back into the U.S., and one of the things on there was like, do you have any snails on you, in part or in whole? And it's because there are invasive snails all over the place. One of them we very purposefully introduced. So the African land snail has been introduced all around the world because it's a tasty thing to eat. But it turns out that African land snails eat everything. So when you introduce this animal, you're like, yay, I get to eat this animal, and then they escape or get purposefully released, and now there are African land snails all over the place decimating habitats. So that's also true. They're tasty, but vicious.
So do we have any questions from our audience about snails? I see 1, 2, 3. I see several. So let's get our question mic over there. I don't know where else we could go first.
Oh, sorry. Oh, hi. I have so many questions. Jan, thank you so much. You answered the Hood Canal question. I want to know why the snail you did the dissertation on is the most awesome. And I also want to know why you sing to snails and they come out.
Oh, okay. So why did I do my dissertation on Cetarium pica, the West Indian top snail, or any of its other 40 common names? So this particular species of snail lives throughout the neotropical Western Atlantic. That's a big fancy way of saying in the Caribbean and Bermuda and all around the Caribbean Sea. And, well, it's the second largest snail in the Caribbean. I bet you all can think in your minds what the largest is, and a really well-known fishery that a lot of people eat fritters when you go to the Caribbean. But this species that I studied is in the intertidal. So we're seeing the conch decline in the Caribbean, and this other species was starting to take its place on menus, and nobody knew that was happening because a snail's a snail's a snail when you pound it and fry it. So, but it's intertidal, so it could get wiped out. So I studied it because I wanted to protect it. It's also just, I don't know, when you study something for 6 years, it becomes like it's like a child to you in a way. Because I literally, why do you sing to snails and they come out? I kind of had to massage their shell in a way because I needed to get them out of their shell so I could take a tissue sample and study their population genetics. I didn't sing to my snails, but I did kind of have to gently hold them a certain way so that they would come out and say hi.
It's funny you mentioned that you spent so much time with that snail. You had a different kind of relationship with them. So one of the things that we work with at the Seattle Aquarium is we talk a lot about empathy. How can empathy better connect people to animals, better connect people to conservation? And one of the sort of the characteristics that help engender empathy for animals and people is continuity. The more time you spend with an animal, the more you feel like you can understand that animal, you can understand its perspective. It's why we think we know what our pets are thinking. And you spend 6 years with a snail, you get pretty close.
Yes, it's true.
Fair.
It made it really hard to go to restaurants. I couldn't eat it.
So we had a couple of questions up here while Michaela is moving. I wanted to ask Eileen, someone was asking about the tubes that you outplant. Do you just leave them there? Do you go back and get them? Is PVC okay to be out in the water?
No, long term. We do go back and get them. So part of the reason we bury them, like in that photo, under so many rocks, it's in part because the abalone live in pretty high current areas, so they'd get rolled around and squished by their own tube. But it's also so that we can come back and get them. So generally, we're actually coming back probably 10 months later to get them. And at that point, all the abalone have crawled out, so you're not worried about crushing them as you move the tube out. I've been doing some other projects for my dissertation recently that have gotten us out there more often. So we've been able to pull them out of the water after about 2 months, and all the abalone have already moved out. But they stick around long enough for us to come get them and then use them the next year. So it's convenient for us too.
So how long do you put a tube of abalone out there, and there's a whole bunch of abalone all together in the same place. That must be super exciting for things that eat abalone. How do you deal with that?
Yeah, we've thought and worried about it. The past several years, Washington Department of Fish and Wildlife, who's one of the huge collaborators on this project, and PSRF have put out time-lapse cameras sort of facing the entrance of the tubes, and they have these time-lapse cameras they've put them facing empty tubes as well as sort of a control. And in all of the photos that we've gone through, we have yet to see a predator with an abalone in its mouth. I think that there's something to be said for like if an octopus wandered by and found 100 abalone in a tube, they might eat them, but we have no evidence of it being like a common thing. It's not like there's fish waiting there for us to put the tubes out.
They're just waiting with the
Yeah.
boat coming in. There's a whole, yeah.
It happens once a year. Yeah.
Question from the audience.
Hi. This is a question for Jen, the photographer. Thank you for the beautiful pictures around moon snails. And so I think I got that one of them was a Lewis, but that's a type of moon snail. So that was one kind of minor question. But so my daughter had a overnight first grade field trip in Indianola. This just a month or two ago, right before end of school. And we thought these things might be garbage that were on the beach. And so we found out they weren't. And that was really cool. And, you know, I'm third generation from Pacific Northwest, had never known about moon snails, felt pretty ignorant about it. And I was like, wow. So very cool that we're here tonight to be able to learn more about moon snails. And I think my question was just about what role does the garbage play? Like, does it dissolve into sand, or like what maybe those things that look like parts of a tire almost when you're walking on the beach? And then my daughter had a question about, I think just the moon snails themselves, like you said, where we see them, like what do they eat, and do they need to be protected, or are they endangered, and that kind of thing.
Those are all really great questions. So firstly, our moon snails are not endangered, so that's really great. I'm happy that I can talk about a species that's not endangered up here. They're thriving and doing well. They love to eat clams. That's their favorite food, and that's why they like to live in the sand. So that's where they find food underneath the sand, are all those delicious clams. And then those egg collars just will disintegrate after the babies hatch out. So all that material, that sand, that mucus, the jelly mass that the eggs were in, that all goes back into the ecosystem, into our marine environment. Did I answer all your questions? Okay, good.
So Jan, I know we'll have a couple other questions. I want to get one from online. Someone was curious about, like, how do we help pteropods? Like for saving other species, we might say we're going to have a harvest ban, or we may do some marine protected areas. But this is a pretty special case for these animals. So how can we help pteropods?
Yeah, that's a really important question, and I alluded to it at the end of my talk. Ocean acidification is caused by one thing, and it's the excess of carbon dioxide in our atmosphere from fossil fuel combustion and from deforestation. So it comes down to, obviously there's making personal choices. That's really important. There's also talking to your neighbors and family. That's really important too, because spreading that message. Most people want to do the right thing, but how many people know about pteropods? They're like a pencil dot, right? And they're out in the water, so they're not very viewable. And then, it's thinking about where our nation goes for energy, and also on a global sense, how we can work together to really affect that. So, unfortunately, it's a large answer that's not so tractable, but I think the main thing is talking to other people that may not know about it and doing it, like in a non-confrontational way. Hey, I heard about this, and that's also affecting oysters, and those are good to eat, It's amazing how much attention you can get when you start talking about food. People really like that.
For sure. I mean, it's about reducing our carbon footprints, right? And for a while, we talked about polar bears and that kind of thing, but pteropods are part of that same mix.
Yeah, and in fact, they indirectly can feed polar bears. You can draw that Arctic food web to the pteropod, then to the fish, and then to the seal, and then to the polar bear. So it's all connected.
Indeed. We had another question, I think, up front here.
I have a really simple question that I think anybody on the stage can answer. Is a slug a snail without a shell?
So you think it's an
you
easy
opened that can of
question.
worms. I think you're going to have to answer it.
But you look at the phylogeny, fancy word for family tree, of gastropods, and the shell has been lost and gained many times. So, kind of. I mean, a slug is a snail that lost its shell, or is a snail a slug that gained a shell? Both are true.
Both yes and.
Okay. That's not true. There are people that definitely eat slugs. Absolutely. I mean, they're basically the same thing as eating a snail.
Yeah, less prep.
Less
right?
prep.
Yeah.
It's so much easier. I mean, if you've ever tried to get a conch out of its shell, it is a task.
Jen, what are some other snails we can find in Puget Sound beaches?
We have amazing whelks. We have dog whelks, dog winkles. They're a pretty common snail we see out there. They lay lots of eggs in the intertidal that many of you may have seen. They can cover entire rocks. They look like they're called sea oats. They're little tiny yellow capsules that we find everywhere. Let's see, what other snails do we have? We have periwinkle snails. Trying to think of some other snails we have. Wrinkled amphissas, we have so many. If you were to go to our field guide, the new Beachcombers field guide is a great one. If you want a book to help you identify things in the intertidal, and if you were to look at that, you'll see pages and pages of snails here that are here in the Pacific Northwest. So I can only scratch the surface of how many are out there.
And of course, the snails are the source of so many of those seashells that people love to collect. And like one of those sort of key messages we're always trying to help with people is to check, leave it, A, leave it. B, if you must, make sure it's empty.
It's really hard to find shells that are not only empty, but don't have another animal living on them. So we do tell folks to leave everything on the beach that's there, unless it's trash, because not only could it be a home for a hermit crab, but it could also have some barnacles living on it, or a limpet, or some algae. All these things use shells in the intertidal for their homes, and when those shells disintegrate, they're putting all of that wonderful building material back out into the marine environment, which is really important too.
So we're nearing the end of our program. I'm wondering if any of you all have any sort of, like your favorite snail facts that you've been keeping in your back pockets, you've been waiting for someone to ask you about. What do you need to tell people about snails?
My whole talk was that.
Good question. So she was asking about different species of abalone, because ours are rather petite, are they not, compared to some other ones?
They are, yeah. No, pinto abalone actually have the longest range of any of them in the U.S., though. They go from southeast Alaska all the way down to Baja California and Mexico. Yeah, and all the others in the U.S. have just names after colors, so black, pink, red, white, green, pinto.
It's way more
clearly
creative.
the best. But yes, there are different species. All of their shells look different. I personally am not great at telling them all apart, but one of my coworkers who was in California with all of the different species can really well. They live at different depths. I'd say red abalone are probably the most well-known. They get really big, arguably the biggest in the world, and they were the ones that were most recently eaten in California, although the fishery for them has just closed recently too.
All right. Oh, you got one. I see it.
So it's not actually a fact, but it's, I
It's
know.
a rumor?
The professor
opinion?
actually
Okay.
has a heart. So, Jen, the moon snails, like I said, I grew up in West Seattle, and those shells fascinate me so much. And you know what fascinates me the most is that blue dot at the top of their whorl, right? It's a darker dot. And what's the most famous blue dot? So I thought that was always interesting that it's called a moon snail, but to me it always looked like there was an earth on top.
That's so cool. I never even thought about that. I love that.
And I love that as an ending note. Thank you all to our speakers. The way we do it in Lightning Talks is we put a applause emoji in the chat, but because we have this live studio audience, our speakers were all amazing tonight. You all were amazing tonight. Thank you all so much for being here. Now we're excited to wrap up. Well, not that excited. We're wrapping up season 3, and we're excited about season 4. So our next Lightning Talks will be in November, and our topic will be ocean technology. So that'll be the first Lightning Talks event of season 4. If you have ideas, please, please, please do send us ideas. It's not like we're out of ideas, but we want to know what you all want to hear about the ocean, about its conservation, about how we value it, about our culture. There's just an infinite, sort of variety of potential topics, and we really want to know what you all want to know more about. Now, if you enjoyed tonight's program, please do consider a donation. It helps keep programs like Lightning Talks accessible and available for everybody. It's a great evening. If you're here locally, please do come down and see us. And don't forget, all of our previous Lightning Talks are available on our YouTube channel. If you're there, please do click like and subscribe. Y'all just go home, click like and subscribe. It helps us, and it keeps Lightning Talks super visible. This has been a great evening. We've had a really good time. Thank you for being here, and have a great evening.